welcome to The Print. This is Akanksha Mishra and you're watching Scientifix, where I will be taking you through this week's top science news from across the world. Our first story today answers a curious question about how bats can carry deadly viruses like coronavirus and yet stay healthy themselves. A study by US scientists at the Centers for Disease Control and Prevention found that Vesper bats, a family with more than 500 species, have two separate sets of genes that produce antibodies, unlike every other mammal which has just one set of these genes. What are antibodies? They are the proteins made by our immune system to protect us from bacteria and viruses. By having two genes that produce them, bats have a potentially greater ability to generate diverse antibodies and respond to infections than other mammals. The discovery not only challenges the current understanding of mammalian immune systems, but it could also help explain the evolution of bats. It might not fully explain why bats are effective reservoirs for viruses, but it does offer a new direction for studying how they tolerate viruses without severe illness. Next up, a new Nature study finds that climate change is not simply increasing malaria everywhere. It is actually shifting where the disease poses the greatest threat. In today's hottest malaria hotspots in West Africa, temperatures could eventually rise so high that malaria-carrying mosquitoes won't survive in them. It would reduce malaria cases by around 5% in countries such as Nigeria, Burkina Faso, and Mali, potentially eliminating the disease in some extremely hot Sahelian regions. But cooler areas, including southern Africa and the highlands of East Africa, could see malaria risk rise as temperatures become more suitable for mosquitoes. By 2100, cases could increase by up to 20% in parts of the Rift Valley and coastal southern Africa. The researchers say that climate mitigation and adaptation will both be essential. Limiting warming to 2 degrees Celsius could prevent millions of infections, while health systems must prepare for malaria to emerge in regions with less experience managing the disease. Next up, researchers at Colorado State University have developed a process that turns carbon dioxide into strong recyclable plastics, which gives an alternative to conventional petroleum-based materials. The process uses a catalyst to combine CO2 with a reactive molecule, which is called bicycloalkane, which creates synthetic plastics that can be made either hard and durable or soft and more flexible. The materials can also withstand heat and chemical damage, while their basic building blocks can be recovered and reused after the plastic reaches the end of its life. This research aims to support a circular economy by using captured CO2 as a raw material rather than releasing it into the atmosphere or simply storing it, because it's abundant, inexpensive, and readily available. The approach could potentially help reduce plastic waste while creating a new use for captured carbon emissions. Finally, DNA recovered from two mummified indigenous individuals in northern Chile has revealed the earliest known smallpox virus strain found in the Americas, providing direct genetic evidence that the disease arrived with European colonization. The individuals lived between 1492 and 1631, and they both have nearly identical viral smallpox genomes, suggesting that they were infected during the same outbreak or by the same circulating strain, which researchers have named the CAM9 lineage. The scientists compared this ancient genome strain with older European and modern smallpox viruses. The researchers found that CAM9 sits evolutionarily between medieval European strains and the lineages that eventually led to modern smallpox. This supports the long-standing view that European colonization introduced the disease to the Americas, where it devastated indigenous populations who had no prior immunity to this disease. The study also reveals how the virus evolved during the colonial period, including changes in its genes and its mutation rates. That's all we have for you today. Thank you for tuning into The Print.